What's the most important organ in your body?
Your heart.
Brain.
Your heart ain't an organ.
Your brain.
Is it?
Your lung.
Yeah.
Your brain. Your brain.
Connor, if your heart was dead, could you still live?
No.
Are there any situations, Jake, where if your heart's not pumping, you can still live?
Yeah, you can. If you get pumped by someone else, like by a machine, get pumped.
Yeah, you can replace the heart
But you can't
with a
replace the
pump,
brain.
but you can't replace your brain. The brain is the most important organ in the body because it controls all the other organs. Now, these two scientists, Charlie Scott, who's from the scientific centre down the road that I talked about earlier this week, the National Institute of Medical Research, and Christoph are doing research on how the brain grows, all right? And what we're going to do today is to look at the cells which make the brain. What I want you to think about is when we're first conceived, how many cells are we? One cell. Angelique, where does the cell come from? From the egg and the sperm. Right, okay. And then it divides into 2 and then to 4. Now, how many types of cell have you got in your body? Name a few. Connor, how many cell types do you think you've got in your body?
3. No, 2. Red and white. Or is that just blood cells?
What about your skin? Are they cells?
Skin cells, yeah. Maybe 5, 6.
Yeah. What about your muscle?
They're all cells as well. Did I?
What about your gut?
That's muscles as well. So 8.
There are about 220 different cell types in the body, which make up your heart, your liver, all different organs. And the brain has how many cell types, do you think? How many cell types? 3 main ones, aren't there? Yeah. So, who knows what nerve cells are called? Nerve cells in the brain. Yeah.
There's another name for them.
I think we'll get the embryos out.
Do you know what that is?
That's a little baby mouse.
That's a mouse embryo,
It's a
yeah.
little baby, isn't it?
Yeah, before it's born.
So it's still
So it's been
Yeah.
12 and a half
no.
days in its mother's womb. Just 12 and a half days after fertilisation.
Oh my God.
What you're going to examine is a mouse that hasn't been born, all right? This is about 2 weeks after the egg was fertilised.
The blue staining is showing you the whole areas where the neural stem cells are. So stem cells are the cells that all cells from of the whole body are formed from. Neural stem cells mean they're the cells that the whole brain forms from.
So you've got a Petri dish with a lid. You can tip your mouse embryo out into the dish.
It looks really cool.
You can see blue staining all over these embryos. So the blue staining is showing you where the neural stem cells are. So the neural stem cells are the stem cells that form the entire central nervous system. And the central nervous system is the brain and the spinal cord. So along this, during embryogenesis, so you can see in the embryo now the blue. And if you have a look down the microscopes, you'll be able to see blue in the brain.
We just need to explain what embryogenesis is. Embryogenesis is how the embryo is made, all right? As the embryo grows, that's embryogenesis.
But if you look really closely, if you look also near the nose, you'll be able to see whiskers. You'll be able to see the hair follicles. And this is because there are actually stem cells also in the hair follicles.
Guys, why do you think there are more stem cells in the hairs of the mouse's nose than anywhere else? Right, good girl. Because the hair falls out, has to be replaced. So you'll find the most stem cells where cells are being regenerated, where they're regrowing. Yeah.
So in the lab, we culture neural stem cells because we want to ask lots of questions of these neural stem cells. So we want to ask what factors they express, so what they need to be a neural stem cell, and we also want to know what they're responding to. So in the lab, we dissect those areas and we basically dissociate the tissue. So we make it single cells and then we make it form a neurosphere, which we have.
Before we go any further, let's just see how many of you understand or recognize what a stem cell is. What is a stem cell?
The cell in which it's the cell in which, like, all cells are formed in stems, isn't it?
Yeah.
So Anjali's got it right, okay? A stem cell is, if you like, the daddy cell. It's the father cell which will make all the cells in the body, or is capable of making all the cells in the body. And so what Charlie does is to look at the father cells that just make the brain tissue, because as we grow, these cells become more and more specialized and limited. So the cells that you're making or using probably can't make skin very easily, but can make brain really well. And the earlier you go, when you go to the embryo, you go right back to the beginning when the egg is fertilized, then you have a cell which has the most potential for making all kinds of cells. And you need stem cells for what reason? Because you injure yourself, don't you?
All right, to repair.
So when I rub my hands like that, I'm losing skin cells. They've got to be replaced. And the stem cells in my hands will be replacing them. So stem cells are there partly in the repair process in the body, and partly as we grow from the very beginning, as in the case of this mouse embryo. Okay? Charlie.
So when we dissect these areas that contain the neural stem cells, so the cells that are going to make the entire brain and the spinal cord, in culture, they become these.
It's amazing, isn't it?
These things that we call neurospheres.
all
So
come
these
from stem
neurospheres are spherical aggregates of cells. So they are a ball of cells, essentially. So one neural stem cell will be able to create this entire neurosphere. And this neurosphere is a little bit squashed, but yes, this has just come from one neural stem cell. It's been able to produce hundreds and hundreds of cells, all that are specialized to become brain or spinal cord.
Charlie, how long does it take to make a ball of cells like that?
So in the embryo, we see them after about 3 days, but to get a big ball like that, it's about a week. So it's very fast.
How many cells will be in the ball?
There are about 10,000 cells.
10,000 cells.
Yeah.
So do you see that's one of the very important issues that that in a normal person, these are nerve cells from the brain, but the normal person can regenerate cells very, very quickly.
How many brain cells do you have?
Okay.
About 3 billion. 3 billion.
And this, on this slide, with this microscope.
3 billion brain cells.
Look, even with a microscope, have a look.
Is that there?
So that's the ball of cells. So in just 3 days, it's made 10,000 cells.
In 3 days? Wow. Well, they kind of look like cotton wool.
Yes, they do.
And what do you do with them brain cells?
Well, we'll study them to see how you can form new brain tissue.
Oh my god. So how do you, what do you get from them?
Well, you ask Charlie that, what she's doing, but what she's doing is she's looking at the genes which help that part of the brain to grow. We thought it might be interesting for you to take some nerve cells, which we're going to look at next. The cells in the brain with which you think are called neurons, all right? And you've got about 3 billion neurons in your brain. And these are constantly being replaced if they die. That's why we are able to continue, some of us, to think for longer. Now, we're going to look at some nerve cells. On slide 3A, you'll find some orange-looking cells. They look something like that. You've got 3 billion cells like that. Those are the things that with which you can see, think, smell, fall in love.
In the culture dish, in the lab, what we do is we take these neurospheres and we make them mature because we want to see what kind of cells they can make. And
How do you make them
sometimes we make them mature. So they have things called growth factors that are making them proliferate. They're making them divide and make more cells. So we just take that away.
Okay.
And we give it something to attach to. So the neurospheres are floating around in the culture dish, but if we stick them onto something and we take away their factors that make them divide and make more cells, we can see what they mature into. And the three cell types of the central nervous system are neurons, astrocytes, and oligodendrocytes. So neurons are basically your nerve cells. They're the electrical cables in your brain, firing, and they're why we can do everything. The glial cells are really there to support the neurons. So when you're focusing, see if you can see the brown splodge, and try and put the light over the brown splodge. Ooh. So once you've looked at the neurons, have a little look at 3B. What is it? 3B is called an astrocyte. So an astrocyte, they're called because they look like stars. So basically they're there for support for the neurons. So they give food and nutrients to the neurons, and they also give the neurons something to grow on and attach to. So they basically give the structure for the entire brain. Have a little look.
If you look here, you can see.
Oh, bloody hell, why did you do
Can
that?
I see? Can I have a picture?
So what our focus is just. So what you see this violet staining is to label the cells,
magnificent,
the neurons in
So
an
what's that
adult.
for?
It's an adult brain. So we will talk about
is
adult
it?
stem cell. Adult. So it's
Oh, it's an adult brain
It's an
cell.
adult brain.
That's just like mine then.
That we have cut that way.
Okay, have a look down there.
I want to say star shape. I see a big circle blob with lines coming out of it.
So all the lines coming out of it is what scientists, if you look on a really high power image, it actually kind of looks like a star. It's not exactly a star, it just looks roughly star-like. So these are the cells that provide the structure to your brain and provide the place for the neurons to grow, and they give nutrients to the neurons.
You look smart for being so small.
Okay, for this next bit, we're going to stain an adult brain. Everybody will need a lab coat on, unless you want all of your clothes to get stained. And you need gloves. This is a slide showing a section of an adult mouse brain. Do you want to have a look what an adult mouse brain looks like? This is an adult mouse brain. This is what you're looking at sections of right now.
So can you pay attention? You've all got some adult brain, and we're going to stain it to see if we can see what are the nerve cells that we're most interested in called? Well, the stem cells, yes, but we're not looking at stem cells, we're looking at neurons. Okay, fine.
So now very carefully pipette some of the stain into the cheap pipettes you've got. You should have a tube with some water in there, just to get rid of the excess stain.
That's great. Brilliant.
I've done it.
I'm going to put it on the top microscope. Guys, this is Jordel's stain slide, and you can see literally thousands of cells with the nucleus of the cell stained dark blue. Remember the part that controls the cells working.
Oh my god, I can see the mouse's brain.
Oh my god!
So if you just watch me first, and then we'll have a go.
Jamal.
So these have all been humanely
Okay, go
killed.
on. Your favourite things in the world?
Oh, again?
Yeah, the little one.
These are mice, not rats. So to start off,
That's the mice, the rats.
Do you got water in it?
it's ethanol, and this is to just keep
That's
away.
just alcohol,
Yeah.
70% alcohol.
So what, I can drink this and get drunk?
Oh my god! She just cut off his head. Oh, look at his little
Doesn't it remind you of Ratatouille?
They just cut off his
Can I
head.
eat it?
Can
I eat
Don't
it?
say that.
Can I eat it?
You can eat this bit.
Yeah, go on then.
Don't
So the
touch
first thing we
it.
do is to try and get rid
I
of
don't
the
want to look at that.
Oh. Yeah, they just cut his head off.
There was a light. Can you plug in the light?
What can I do? I'll be dead.
So now we have to factor in the skull, which can be very difficult. So first of all, we've got rid of all the ears and the skin around there.
It's there for like 10 years.
So then how we cut the skull is to go with the scissors up like this, so we're not hurting the brain. So you try and point it up.
But alright, Charlie.
Yeah,
Is that better?
much better.
Move it over to there where there's more light. That's better.
Hey,
Oh,
you,
now
you
you
can
do.
read the projector away now, we got proper light.
We have to try and peel
Read
back
the
the
projector.
skull.
It's absolutely revolting. Hey, put the projector down, we got proper light now. We don't need no more, just interrupt them.
So we start peeling back the skull, and you can see the brain now.
Because there's frontal lobes.
You can see us here as well. Can you see here? These bits here. These are called the olfactory bulbs. Because to a mouse, smell is really, really important. So that's why the olfactory bulbs are quite big.
We have
What is it called? A fungi punch?
Olfactory bulbs. Olfactory bulbs.
Perhaps you should hold the scissors because I'm worried about cutting you.
The skin that way.
Yeah.
So you cut out the ear.
Could be already done it.
Yeah, you look done it.
That's brain surgery right there.
It's a business.
Fantastic. Well done. Well done, girls. Terrific. Guys, I forgot to tell you it's brain for lunch, all right?